Today we will discuss about chemiosmosis, that is also called chemiosmotic hypothesis. Now, chemiosmosis or chemiosmotic hypothesis is given for the production of ATP. Okay, it is given for the production of ATP. In the last class, we discussed about the light reaction, in which we observed that the light reaction is taking place in the thylakoids and it results in the production of ATP and NADPH. Now we have done that ATP is formed in the light reaction. But how that ATP is actually formed? We just discussed that when the electrons are coming back from higher to the lower energy state, the energy will be released and that energy is actually utilized for making ATP, right? I'll just give you a view of light reaction. Okay, so here we saw that when the electrons are coming back from higher to the lower energy state, energy is released and that energy is actually utilized for production of ATP. But how this ATP is getting formed? This is not a single step in which the ATP is getting generated, but we have series of reactions for that. So chemiosmotic hypothesis will explain the process of ATP production, that how ATP is synthesized. Now there are some important requirements for this hypothesis, and these requirements are thylakoid membrane. Okay, so for the ATP production we need thylakoid membrane, then we need protons or hydrogen ions. Apart from this, we also need proton gradient. Gradient refers to the difference. So proton gradient is the difference in the concentration of the protons. Then apart from this, we need ATP synthase enzyme, which helps in the production of ATP. So these are the requirements for the chemiosmotic hypothesis. Now I am going to draw a diagram. This is the thylakoid membrane. We have already done the structure of chloroplast, right? In chloroplast, we discussed that there are small rounded structures which are called as thylakoids, and the group of these thylakoids is called as granum, right? So I have drawn here thylakoid. Okay, so this is a zoom view of the thylakoid. This is the thylakoid membrane, outer membrane, inner membrane of the thylakoid, and the space inside the thylakoid, this space, that is called as lumen of the thylakoid. Okay, just like here, this is the membrane and this space, okay, this space is lumen. Now we already discussed the light reaction that is taking place in the thylakoid membrane, which involves photosystem 2, photosystem 1 and then electron acceptors, right? So here we have photosystem 2 that is embedded in the thylakoid membrane and it absorbs at 680 nanometer wavelength. Now when light energy falls on the photosystem 2, it results in the breakdown of water in the presence of light. That is called photolysis of water. So water will be broken down and it is converted into hydrogen ions, oxygen and electrons. These electrons will be given to photosystem 2, and the electrons from photosystem 2 will be given to plastoquinone. Okay, PQ is the plastoquinone. That is your electron acceptor. Okay, so we have number of electron acceptor molecules which are embedded here in the thylakoid membrane. So you have PQ, that is plastoquinone. When the plastoquinone accepts the hydrogen ions and electron, it will be reduced and it will be converted into PQH2, that is reduced plastoquinone. Okay, so it is converted into PQH2. ये reduce हो गया because it has accepted the electrons coming from the photosystem 2. Now once the electrons from photosystem 2 are given to plastoquinone, during this step the hydrogen is pumped into the lumen of the thylakoid. Now what is this outside space? This outside space is the stroma, that is cytoplasm of the chloroplast. I'll draw the structure of chloroplast again. This is the outer membrane of chloroplast. This is the inner membrane of chloroplast. Here we have thylakoids which are piled one above the another to form granum. And all these granum or thylakoids are interconnected by membranous extensions which are called as stromal lamellae, right? Then this part, this dotted part, represents the cytoplasm of the chloroplast, that is the stroma. Okay, so in this diagram I have drawn the thylakoid and its outer part, that is stroma. So what happens when the electrons are being passed from photosystem 2 to plastoquinone, that is electron acceptor? Then while passing of electrons, the hydrogen from the stroma will be pumped into the lumen of the thylakoid. Now this plastoquinone, that is PQH2, reduced plastoquinone, will pass its electron to the another electron acceptor, that is cytochrome B6F. Okay, cytochrome B6F. Then the electrons from the cytochrome B6F will be given to plastocyanin, PC. Okay, PC stands for plastocyanin, and this plastocyanin will give its electrons to photosystem 1, which absorbs at 700 nanometer. Now once the electrons are coming from plastocyanin to the photosystem 1, energy is going to be released, right? And that energy here is utilized for making NADPH. So how NADPH is formed? NADP is present in the stroma. It will accept the protons from the stroma and it will be converted into NADPH. Now you will see कि स्ट्रोमा जो है दैट इज साइटोप्लाज्म ऑफ द क्लोरोप्लास्ट, वहाँ पे हाइड्रोजन आयंस की जो कंसंट्रेशन है दैट इज लेस एज कंपेयर टू द ल्यूमन ऑफ द थैलेक्वाइड. ल्यूमन के अंदर हाइड्रोजन आयंस की कंसंट्रेशन ज़्यादा है. अब ये स्ट्रोमा में हाइड्रोजन कम कैसे हो गए और ल्यूमन में ज़्यादा कैसे हो गए? फर्स्ट स्टेप यहाँ पे देखो. व्हेन फोटोसिस्टम 2 एब्जॉर्ब्स लाइट एनर्जी, तो वाटर की फोटोलिसिस हो रही है और इस वाटर की फोटोलिसिस से वी आर गेटिंग हाइड्रोजन आयंस. तो एक स्टेप तो ये हो गया यहाँ जहाँ पे ल्यूमन के अंदर हाइड्रोजन आयंस बढ़ रहे हैं, राइट. सेकेंड स्टेप, व्हेन इलेक्ट्रॉन्स आर accepted by plastoquinone is reduced, then during this step electrons are pumped from stroma to the lumen. So this is the second step जहाँ पे आपके ल्यूमन के अंदर हाइड्रोजन आयंस आ रहे हैं. तो फर्स्ट स्टेप ये था जहाँ पे वाटर की फोटोलिसिस हुई और ल्यूमन में हाइड्रोजन आयंस एक्यूमुलेट हो गए. सेकेंड स्टेप जहाँ पे प्लास्टोक्विनॉन रिड्यूस हुआ एंड वाइल इट्स रिडक्शन इसने हाइड्रोजन आयंस को पंप किया फ्रॉम द स्ट्रोमा टू द ल्यूमन. तो यहाँ पे भी हाइड्रोजन आयंस एक्यूमुलेट हो गए ल्यूमन के अंदर. तो इसका मतलब ल्यूमन के अंदर हाइड्रोजन आयंस की कंसंट्रेशन बढ़ती जा रही है. और स्ट्रोमा में कैसे कम हो रही है? बिकॉज ये जो हाइड्रोजन आयन है दैट इज बीइंग पम्प्ड फ्रॉम स्ट्रोमा टू द ल्यूमन. प्लास्टोक्विनॉन ने स्ट्रोमा से हाइड्रोजन को ल्यूमन के अंदर भेज दिया. तो स्ट्रोमा में हाइड्रोजन आयंस कम हो रहे हैं, राइट. और स्ट्रोमा में कैसे हाइड्रोजन आयन कम होगा? बाय द प्रोडक्शन ऑफ NADPH. NADPH की फॉर्मेशन के लिए आपको जो प्रोटॉन्स चाहिए दे आर बीइंग यूटिलाइज्ड फ्रॉम द स्ट्रोमा. सो इट मीन्स स्ट्रोमा में हाइड्रोजन आयंस की कंसंट्रेशन कम होती जाएगी एंड ल्यूमन में हाइड्रोजन आयन की कंसंट्रेशन बढ़ती जाएगी. एंड इट हैज़ रिजल्टेड इन द प्रोटोन ग्रेडियंट. देयर इज डिफरेंस इन द कंसंट्रेशन ऑफ द प्रोटोन्स. स्ट्रोमा में लो हो गए और ल्यूमन में हाई हो गए. नाउ व्हाट विल हैपन? पैसिव ट्रांसपोर्टेशन विल टेक प्लेस. पैसिव ट्रांसपोर्टेशन मीन्स movement of substances from the region of high concentration to the region of low concentration. तो ल्यूमन में हमारे पास हाई कंसंट्रेशन है हाइड्रोजन आयंस की एंड स्ट्रोमा में लो है, राइट. सो हाइड्रोजन आयंस विल स्टार्ट मूविंग फ्रॉम ल्यूमन टू द स्ट्रोमा, दैट इज फ्रॉम हाई कंसंट्रेशन टू द लो कंसंट्रेशन. नाउ हाउ दे विल कम आउट ऑफ द ल्यूमन इन द स्ट्रोमा? For that we have another protein which is present in the thylakoid membrane that is called as ATP synthase. ATP synthase. And this ATP synthase has two subunits. This one is CF naught which is embedded in the membrane. And then we have CF1 which is outside of the membrane. Okay, it is located towards the stroma. So it will provide a channel through which these hydrogen ions can be transferred from the lumen to the stroma. So hydrogen ions lumen में accumulate हो चुके हैं. Now they will pass through this ATP synthase. Okay, they will come in the stroma. And for each hydrogen ion being passed from lumen to the stroma, you will get 1 ATP. Okay, when the protons or the hydrogen ions are being pumped from lumen to the stroma, energy is going to be released. Okay, जब भी प्रोटॉन जो है वो अपनी हाई कंसंट्रेशन से लो कंसंट्रेशन की तरफ जाएगा थ्रू द ATP synthase, तो एनर्जी रिलीज होगी and that energy is actually utilized for making of ATP. So this is how the ATP is generated in the light reaction. I'll repeat again. You have thylakoid membrane and you have stroma, right? In the thylakoid membrane you have photosystem 2. फोटोसिस्टम 2 पे जब लाइट फॉल करेगी इसके इलेक्ट्रॉन एक्साइट होंगे, दे विल जंप फ्रॉम लोअर टू दी हाई एनर्जी स्टेट and they will be accepted by the electron acceptor, that is plastoquinone. While the electrons are accepted by plastoquinone, the hydrogen from the stroma will be pumped into the lumen. तो एक तो ये स्टेप हो गया जहाँ से ल्यूमन के अंदर हाइड्रोजन आ रहा है, एक यहाँ वाटर की फोटोलिसिस भी हो रही है, उससे भी यहाँ पे हाइड्रोजन अंदर एक्यूमुलेट होता जाएगा, तो ल्यूमन में हाइड्रोजन की कंसंट्रेशन बढ़ रही है. These electrons are then given to another electron acceptors like cytochrome B6F, plastocyanin, and then from plastocyanin the electron is given to photosystem 1, and this photosystem 1 will use the energy of electrons for the synthesis of NADPH. And again, for the production of NADPH, the hydrogen ions are utilized from the stroma, which will further decrease the concentration of hydrogen ion in the stroma. So you can see in the stroma the concentration of hydrogen ions is decreasing, while in lumen it's increasing. So we have concentration gradient, okay, difference in the concentration of the protons, as a result of which protons will start moving from high concentration to the low concentration, okay, that is they will start moving from lumen to the stroma, and this movement will take place through the ATP synthase, which has two subunits, CF0 and CF1. CF0 is embedded in the membrane, while CF1 is located towards stromal side. So for each proton which is being pumped from lumen to the stroma, one ATP molecule will be synthesized. While the proton is coming from lumen to the stroma, energy is going to be released, and that energy is utilized for the production of ATP. So this is your chemiosmotic hypothesis for the production of ATP. And with this we have completed our light reaction. In the next class we will discuss about dark reaction, in which we will discuss that how the glucose is synthesized by using the ATP and NADPH, which are synthesized in the light reaction. That's all for today. Bye bye. Take care.